Greetings, my name is Chiara Cardona. I am a graduate student from Dr. Cristina Velasquez Laboratory at the Institute of Neurobiology, and I will be presenting to you one of my projects titled Effects of BPA Exposure on Anxiety Behaviors Necessary for Survival. I will start with an introduction of this important chemical. Bisphenol-A acts as an anthropogenic stressor that is mass-produced to assist in the manufacturing of almost all plastics and resins since the 1960s. It is commonly found in the air, dust, food, and water. The primary route of exposure throughout the population is through oral exposure because BPA is found in almost all of plastics that we have and we use. The Environmental Protection Agency has declared this chemical as the third highest priority environmental hazard. The concentration in the environment of bisphenol-A varies depending on its location, its temperature, pH, source, and time of sampling. The current concentration that have some adverse effects within the central nervous system, as declared by EPA, is 50 milligrams per kg. Plastic pollution has become an increasing concern over the past 6 decades, with BPA production surpassing 7 billion kilograms per year. BPA have some effects in the central nervous system and, as consequence, in our behavior. Previous studies have shown that at low BPA concentrations, as 2 milligrams per kilogram, have some neurotoxic effects in brain and behavior. Animal studies have linked this chemical to anxiety, aggression, impaired brain development, poor learning, and social interactions. Early developmental exposure to this chemical produces increasing sensitivity towards stress and the risk of developing anxiety behaviors, and increases FKBP5 gene expression in the brain. This protein, FKBP5, is an important modulator of stress in our central nervous system. Current research has determined a link between BPA-induced epigenetic changes in early childhood, leading to altered expression and DNA methylation of this protein, which is associated, as I said earlier, to increasing sensitivity towards stress and the likelihood of developing PTSD and anxiety behaviors. The gap in knowledge is relevant to answer this question. Does BPA exposure, including the 50 parts per million concentration, that is, the gap in knowledge is relevant to answer this question. Does BPA exposure, including 50 parts per million, that is the current lowest observed adverse effects concentration by the Environmental Protection Agency, affects contextual fear extinction learning, which is the paradigm used to study anxiety behaviors? Our hypothesis states that early exposure to this chemical increases the likelihood of developing fear extinction deficits correlated to anxiety-like disorders. This project has principally two aims. One is to determine if BPA exposure elicits contextual fear extinction deficits in adolescent male mice, and the other one is to determine if this chemical elicits the expression of the FKBP5 gene in the same mice. Selected a hippocampus-dependent learning paradigm, contextual fear conditioning and extinction, based on previous studies, because in those studies have been shown a decrease in the number of neurons within the hippocampus and an enhanced persistent fear memory in response to the exposure of BPA. However, no extinction learning experiments have been done to establish correlations with anxiety disorders. The experimental design is as follows. I exposed the mice throughout 14 consecutive days to different concentrations of BPA. These concentrations are 50 parts per million, 20 parts per million, and 2 parts per million. After the 14 consecutive days of BPA exposure, the mice underwent 5 trials of fear conditioning learning. On those trials, they received 3 food shocks between intervals of 90 seconds. After the 5 trials of conditioning, the mice underwent the fear extinction learning phase. In here, there is no presence of the unconditional stimulus known as the food shock, and in here also the trial is 410 seconds. Here is the timeline of my project. First, as I said, I start with the BPA exposure throughout day 1 to 14. Within the days 15 and 17, the mice underwent the fear conditioning learning phase with 1 trial in the morning and 1 trial in the afternoon. Within days 18 and 19, the mice underwent the extinction phase learning. On this graph, we have the BPA exposure and its consumption. In a dark green color, the control group, and in light green, the 50 parts per million mice. In yellow, we have the mice that consume the solution with 2 parts per million, and in light blue color, the mice that consume the solution of BPA with 20 parts per million. Overall, within the experimental groups, we could see how the pattern is basically the same after the day 5 through the day 14 of exposure, and also we could see how the control group is statistically significant more different than the experimental groups that consume the BPA solution. On this slide, I present to you 2 videos of the behavioral test. In the first one, we have the mouse in a conditioning trial. We could see how it explores the cage until the food shock is given. This is a normal response for the fear contextual learning. In this case, he will learn to associate the context, that is the white and black lines in the cage, with the food shock. For the other part, we have the mouse within an extinction phase. In here, the mouse is expected to move and explore the cage more freely than in the conditioning phase. In this case, he has to dissociate the context with the food shock that is not given. If the mouse kept showing freezing response, it is known as an extinction learning deficit. The mouse have extinction learning deficit. Using animes as the tracking software, I was able to create this histogram, which is showing the entire trials and within the freezing time. Each block represents 10 seconds of the entire trial. Green represents 0 seconds of freezing. Yellow represents 0.5 seconds of freezing. And red is encoding 10 seconds of freezing, meaning that in that interval of 10 seconds, the mouse was freezing throughout the 10 seconds. Saying this, we could see that both groups, the experimental group and the control by the second trials, were showing freezing behavior. We could see how practically all groups learn to associate the context with the food shock showing greater freezing response. By the end of all the conditioning trials. In the fear extinction phase, the mice show an interesting pattern of dissociation of the fear learning previously learned from the conditioning phase. All groups within the first trial of extinction expressed a lot of freezing, which is normal because in here they have to learn to dissociate the context from the food shock, which is not given. After the 3 trials of extinction, we could notice how the 50 parts per million exposed mice show and kept showing freezing response. On the contrary, lower concentrations of BPA exposed mice show freezing response. And in the control group, we could see how basically they learn to dissociate the freezing response from the context. Overall, we could see how the 50 parts per million group kept showing freezing response, meaning that those mice have extinction learning deficits. On this graph, we have measured the freezing time in seconds throughout the 10 seconds interval of the entire trial of conditioning phase. In the first trial, we could see how the 2 parts per million mice group show more freezing time than the other groups and is statistically significant towards the control group. In the second trial and in the third trial, we could see how all of the groups have shown more freezing response, but in this case, the 2 parts per million mice group kept showing more freezing response. Overall, in the 5 trials, we could see how they learned to associate the context to the food shock, but in the 5th, we could see how the 50 parts per million mice group show more freezing response than the other groups and is statistically significant, more different than the control group. In the context extinction learning, we could see the pattern that we see on the histograms. We could see how the freezing response in the 50 parts per million group is more than the other groups. Overall, the BPA exposed mice show greatly freezing time that is statistically significant for the freezing time in the control mice group. In the third trial, we could see the learning extinction deficit that we saw on the histograms for the 50 parts per million mice group as compared to the control. In conclusion, the expression of fear in adolescent mice is significantly more in bisphenol-exposed mice than control group during condition and extinction learning. These results provide novel evidence for the capacity of common contaminants such as BPA and physiological relevant concentrations to lower the threshold for neurological disorders in response to challenges, specifically learning in adolescent and adult mice. Our preliminary data with BJ cells further shows changes in FKBP5 gene expression relevant to anxiety behaviors correlated with the chemical exposure. Future experiments in adolescent mice will further address the gap in knowledge of the effects of this chemical at lower concentrations in open field test experiments and the expression of the protein FKBP5 in the brain of the BPA exposed mice before and after this behavioral test. For your attention, if you have any questions, I will gladly address them. Thank you.